Hello, and warm welcome from my side. My name is Dürkop. I'm the Managing Director of Novasign GmbH. Welcome to this session today. Today we're going to talk about digital twins. Before we start talking about digital twins for bioprocessing, I quickly want to introduce Novasign to you. Basically, so Novasign is a spin-off from Boku University in Vienna. So Boku University is a university that has a department for biotechnology since 30 years. And in this department of biotechnology, we always focus on accelerating bioprocess development. And something like 5 to 6 years ago, actually, we thought about how can we make things better and how can we use machine learning in order to accelerate this process even further. Based on that, we founded the company Novasign in 2019 with the aim, as said, to speed up the development process. And we already have quite some customers that use our tools in order to accelerate this development. So now coming to a digital twin, I mean, some of you, including me, might already be fed up by the word digital twin, because digital twin can be anything or nothing. So what is a digital twin? What should it do? And that's often quite unclear. So you have to be quite precise. And when you talk about digital twins, what is actually the aim of a digital twin and what should it do? So in order to shed light in what I understand in the digital twin, or what a digital twin for bioprocess can potentially help you, I want to guide you through with a high-level overview, and then we go into some use cases to get some details about what you can do with these tools and why it will be beneficial to implement such tools in the future. So starting off very easily, you might know this 4P model. So the 4P model basically describes if you have a certain patient that has a certain medical need, this patient needs for sure a certain product. Okay, so there's a request from the patient to the product, and the product for sure has a request for the process, and the process itself has a request for the plant. So basically these are these 4 things that are connected. And if you go in the other direction, basically the plant has to fit the process has to fit the product, and the product has to fit the patient. So easy as that. And currently there are a lot of initiatives for a digital transformation of all of these things. And a lot of companies like Google are investing in the interface between the patients and the product. And other companies, for example, that are more into plant engineering and building up facilities, they are more in the interface between the plant and the process. So now, in order to talk about digital bioprocess or digital twins for bioprocessing, I consider this to be in the interface between the process and the product. So basically what we want to mimic, what we want to create is a digital representation of the interface between the process and the product. So now what should this thing do? It should basically be an in silico representation, as I already said, of exactly this process and product dependency. It should basically help you to understand the relation between your critical process parameters and your critical material attributes to your critical quality attributes. This is what it should help you to understand. And at the end, it must have, and this is a prerequisite of a digital twin, to have a real-time connection to the process itself in order to control it. So that means you have this understanding in a digital twin, and then this digital twin should directly or must directly interfere with your process in order to do something, to control something. And now the question is, what can we actually do? And this I will highlight in the next slides. So what are the potentials of a digital twin? How we at Novasign basically envision a digital twin, it should be in the center. So the digital twin should be in the center and should be a process representation, as I just showed you. So in order to basically train this digital twin to be able to be a digital representation of something, for sure we have to perform experiments. So there are experiments that are performed in early or late stage process development. And from this data, we basically train the digital twin, and then the digital twin should give us process understanding. And from this process understanding, then we take the decision how to set up the process the ideal way. And afterwards, we take in, and that's important, to transfer this knowledge for process control and scale up. So by doing so, the idea is to have a, that the digital twin basically follows the product life cycle of a product. Early stage development, late stage development, and afterwards using the same digital twin for scale up and control. So that is the vision here. From the perspective of what it should deliver to you at the end. So yeah, if we think about early stage development, you can do a lot of different experiments. You can do a lot of changes in your process. And all of these different processes, they lead to different results. So if you plot the response on the y-axis, and let's say the response is, for example, a certain product quality or a certain product amount, you see that depending on how you set up your process, all of these processes lead to different quality, to different titers. So the aim basically is, I'm sorry. The aim basically is to find the process at the end that delivers the highest product quality or the highest product amount. So this would be something where a digital twin can be used in the laboratory, in the early stage development, to exactly identify these ideal processes, ideally in silico, and then you just take this in silico suggestion, perform the experiment, and ideally you end up at exactly that point what the digital twin suggested you. The second thing then, when you think about later in the process, or when you think about using the digital twin for manufacturing purposes, sorry, is about always maintaining the same product quality or the same product amount. So when you think about, okay, this is now our ideal process, we identified this, how do we now operate it always to get the same product outcome? So if you now have a set point process, normally if you perform this process over and over again, you get quite likely the same results. But sometimes it might also happen that you see during your process that we're ending up somewhere completely different. And now the question is, everyone knows when looking at the KPIs in this process right now, okay, something is going wrong, but now the request for the digital twin or the task it should fulfill is basically to bring back this process and to bring back the quality where it should be. So to change the process parameter in order to get back to the desired quality. And this can be seen as a model predictive control approach. And today I will show you both showcases for the first case, for the process optimization, and also for this control purposes. So how can we basically control our process by means of a digital twin? So what can it deliver? As said, different perspective, but same picture. Faster process understanding. So we want to reduce experimental effort to describe a certain design space in the QBD concept. We want to use this process knowledge or these process models during scale up to basically take this learning from the small scale and bring it to the larger scale. And finally, we want to use the type of models for process control purposes. So what I just showed you, okay, something goes wrong. How can we interact with the process in order to get to the desired quality? How is the workflow to deliver such a digital twin? It's actually quite simple, but it has to be clearly defined. So you have to start with a goal. If you talk about digital twins for the sake of talking about digital twins, it's nice to have a buzzword, but at the end you really have to say what is the purpose of the digital twin and what should it do. So if you say, okay, I want to optimize, okay, I want to control, it's a clear purpose. And then you have to say, okay, in which range, with which factors, and so on and so forth. So you really have to clearly define what the digital twin should do. And based on that, you can then start considering the experiments you have to perform. Based on that, you collect data. So all of these experiments, they basically deliver you a lot of data. From this data, you can set up a process model that should describe this data. And then you put this process model in a digital twin where you can do in silico simulations to say what would happen if. So what would happen if we now change the temperature? What would happen if we change the pH, etc., etc.? And the digital twin would basically suggest you new experiments. So you can see this as an iterative loop. You define a goal, you create experiments, you select data from that, you create a process model. The process model is put into the digital twin. Then you get suggestions for ideal process conditions. Oops, wrong button. And then basically the digital twin either suggests you new experiments, or when you're happy with the results, you can basically take this knowledge and, as I showed you before, transfer it for the upscale or use it for the manufacturing directly, depending on where you perform this knowledge. For that purpose, we have developed a hybrid modeling toolbox at Novasign. You can also use it. So if you have your smartphone, you can simply scan this QR code here, and you can start using it for free. So it's a freemium concept. You can start modeling your process with that tool. And if you're interested, you can always get back to us, and we can also help you to set it up the right way. So this basically hybrid modeling toolbox with the digital twin inbuilt helps you to get time-resolved process models, so for upstream and downstream, and it's used for optimization purposes, and you can use it afterwards, as said, for control. And this is what I will show you now in the next 20 minutes. When we talk about hybrid models, that's something you might be unaware of. What is a hybrid model, basically? So a hybrid model does exactly the same task as all of the other models. You have something to describe. So let's say you're interested in describing a critical quality attributes. Let's say we have two here, critical quality attribute 1 and 2, and we want to describe them as a function of several critical process parameters or critical material attributes inputs. The way how it works is that you have two different model structures that are connected somehow with each other. In our case, for the upstream, it's a serial hybrid model. So we have some mechanistic equations that simply describe, for example, cell growth or production of an antibody or production of an AAV or whatever you're looking at in your process. And then we have a machine learning part, in our case, an artificial neural network that describes the rates and the unknowns in these mechanistic equations. And by connecting these two worlds together, you get a very precise identification of your design space with a small number of experiments. You can use this knowledge for process scale up, and as said, due to the structure of the model, and this is what I will show you afterwards, is you can take this type of model and integrate it directly for control purposes. One of the publications we have in that direction is basically a comparison between a pure mechanistic model and a hybrid model to describe a CHO cell culture. And what I want to emphasize here is the difference of how these two type of model works. So when you look at the mechanistic model, a mechanistic model has a strong built backbone where you basically have a certain assumption that defines the model structure, and it's quite independent of the data. And you will see now I have 4 different data distributions, and you will see how the model changes over the different data distributions from the mechanistic model and from the hybrid model. The hybrid model and the mechanistic model here, if you take 4 initial experiments, in this case, in this publication, where the link you can follow down there, we had a 5-dimensional design space with over 100 experiments, and the data points you see in here are from 38 experiments, I guess. You see that if you have initial 4 experiments and you don't really choose this experiment wisely, you see that both models are not really accurate. So if you now choose optimal for experiments, and this is where this loop of process modeling, what I described to you before, comes into play. If you think about how you do this, then you see that the hybrid model already very precisely describes the whole design space. And here, when I talk about design space, the aim of this here is to describe the product formation of an antibody in the CHO cell culture. But when you look at the mechanistic model, you see that basically the model efficiency is very similar to the initial 4 experiments. So that means that the model is as good or as bad from the very first point, and it doesn't actually need any data point, or it doesn't need data in order to be trained. So you see it works very, very similarly. Yeah? And then you can iterate it through. You can say, okay, now here we have 19 experiments, and you see the hybrid model, gets better, but also not a lot better. And the same applies for the mechanistic model. So in a nutshell, at the end, what you can see is if you perform 4 very clever experiments, so in terms of clever, you choose wisely which experiments you do, and you have a model like a hybrid model, you can describe this design space quite accurately. And that means this model covers the nature of the process intrinsically. And then you can use this type of model to simulate all of the different process conditions and optimize your process at the end. So just if you want to look at this again, so I want to emphasize here we have only 4 experiments. Here we have 35 experiments to the right-hand side where you see age. And basically what you see is that you can describe the product formation quite accurately or similarly with both of these data distributions, while on the left-hand side you only performed 4 experiments. Yeah. So you can save a lot of experiments by that in process development. You don't need a lot of prior knowledge because the hybrid model has a rigid structure in the backend, but learns from data straight away, so it can easily be applied. And you see that the description here on this data set is better than the mechanistic model, apart from the first data distribution, I would say. So now when you come about use cases, how can we now apply such a hybrid model or a digital twin in order to describe design spaces? We have several approaches how we can perform experiments and how we can extract this data. So one of the things here I want to emphasize is model-based design of experiments. It's also something we published down there. And what you basically do is you are following this workflow I showed you previously. So you start with a certain objective. Okay, you want to describe space-time yield. You want to describe glycosylation whatsoever. You have a clear aim of what your model should deliver. You start with some initial experiments. You create a hybrid model based on that. The hybrid model is put into the digital twin. And then we can do almost free of charge. We can click the button and simulate thousands, 10,000s of different cultivations in silico. And then the tool tells you what is the best condition to be performed. And then you look at the result, and the result is either a new optimum. So that means the tool predicts a certain process which you did not perform in the lab, and then you can go back, or a certain set of experiments, and you can go back in the lab and then perform it, or to a certain extent at a certain time point, then basically the tool does not predict a new optimum, and then basically you are finished with your optimization process. With that, you can iterate through the design space quite quickly, and you can save a lot of experimental effort. And also, this was already published down there. It's a simple workflow from experiment to data to model to knowledge, because all of that is basically covered in the digital twin, and all of this knowledge is stored. So a new employee can open this tool, look at the process model, can play with it, and can understand, okay, how temperature, how feeding rate, how whatever dissolved oxygen really affects your process in terms of with respect to productivity or glycosylation or whatever is the case is the factor you want to optimize. The other thing I want to show you is something what this is more for late stage processes. So when you are in the larger bioreactor and you want to characterize your process, so to prove in front of the authorities, okay, I have to characterize what's happening around of my optimum, what is happening if I operate at 1 or 2 degrees more or less. How can I basically describe this faster than performing all of these experiments sequentially? So when you look at this, for example, this is this data here is from an E. coli case study from a 20-liter bioreactor, and each of these points represents one experiment. So we have a 3-dimensional design space with 3 levels. That means 3 to the power of 3, 27 experiments. And if you would now do this process characterization on a larger scale, you most likely have only one bioreactor. And that would mean you would take almost like half a year just performing these experiments in order to understand what is happening in there. So it's quite time consuming. The thing we want to suggest here is this intensified design of experiment approach, where you dynamically change process parameters. So you can think about this like you start with one of these points, and then they are connected by these lines, and everything that's connected is basically one experiment. So rather than performing just one point, this one experiment, here we pack something like 3 experiments in a single experiment. So that's what you see here, basically. So we start from the top left corner, go to the center, and then go to the bottom left corner, and this is one experiment. By doing so, you can pack multiple experiments into a single experiment, and by that basically are able to describe the design space much faster. So rather than doing 27 experiments, in this case, we only perform 9 experiments. And now how does this basically unscramble at the end? When you look in the center, or when you look at this graph in the center, let's say where this point cloud is, you see there's a prediction and there's a measurement, and the prediction basically describes what the model predicts you, and the measurement is everything you describe by means of measuring. So, and the point is, if both of these, if all of these points are on the diagonal line, that means your model perfectly describes your data. And what we did here is we created a hybrid model based on these intensified runs. So what I showed you to the left, we changed process parameters dynamically and basically applied this model then on the static conditions. So that means when you now train your model on only 213 observations coming from 9 experiments, you see how this model in orange, or how this data in orange and the model based on that is able to precisely predict what is happening in the test data, which is this greenish triangles. Apart from some of these runs, which are not predicted very well, the majority of the data is really predicted in a very good way. And what this basically tells you is you can use 9 experiments, doing dynamic experiments, and learn from that, and apply this model on 27 static experiments. And the 2 graphs to the right are basically example of this validation data, which is ordinary DOE data, where nothing was changed in the process, and you can see how the model, which was trained on IDOE, can very precisely predict what is happening inside this design space. So in a nutshell, you can significantly save experimental effort for process characterization. And also this was also published. So you see 2 links here. The first one is from this case study, and the second one basically is a publication we just published together with Boehringer Ingelheim, where they performed the same approach in a CHO cell culture, basically. And gives you a similar picture about that. So what does it mean? Yes, you can save up to 70% of experiments, at least how we see this right now. Maybe you can save even more, but this idea is still in a very early shoes. So if you want to try it out, just read the publication and consider how you want to create your models yourself. I think that's a very promising approach for the future to faster learn from experiments, and not just perform static experiments, but really introducing the dynamics, changing processes, and therefore learning more in the development, and maybe also potentially in the future setting up processes like that. I mean, no one says that a process must have constant parameters. You can also change temperature, feeding rates, whatsoever dynamically over the time course if this yields to higher productivity, lower glycosylation whatsoever. It's a very streamlined workflow. You come from experiment to data to model to knowledge, the same as before. And as already said, it's already currently in use also at the first pharmaceutical companies. Okay, now coming to the process control side, how can we now use these models? Not only to optimize an early stage, but also then to use it for process control on the manufacturing side. So here, for example, we have a collaboration with Siemens together in Vienna. So this is a picture of the Siemens Living Lab in Vienna. So you see there's a small bioreactor to the back. And what we wanted to do here is we wanted to create a soft sensor based on a DOE, where we changed pH and temperature. And at the end, the soft sensor should predict the current biomass in the bioreactor without directly measuring the biomass in the bioreactor. And the application for something like this would be to create a soft sensor that says, okay, now you have 2 grams per liter, 3 grams, 6 grams per liter. And based on that, you can then take decisions like induction of cells, transfection of cells, harvesting of cells, whatsoever can be taken. So if you look at how this works, so we characterized the design space, we created a process model, and now we took validation data, so a complete different run. And what you see is how the hybrid model predicts quite accurately what's basically the current biomass concentration in your bioreactor, which is this red line, based on the pH, the temperature, and the current off-gas measurement. And then for validation purposes, we also performed cell dry weight sampling. So we sampled and put it on a balance, and after drying the biomass, and you can see how accurate the soft sensor is. So by that, as said, you get a quite precise prediction of biomass at each time point, and you can take something like that for decision making during a cultivation run. Yeah. The next thing, which is even more advanced, is a model predictive controller. So that means here now really the digital twin comes back and says, okay, now I want to control my process. I learned from the process. I know how input and output is connected. Now I really want to control my process. That is the ultimative goal of a digital twin in the manufacturing side. And here, basically what we did is we took the same showcase I showed you before for this IDOE showcase, so the microbial 20-liter cultivation, and we simulated a pump failure. So now imagine you're in the production side, everything is running smoothly, and suddenly your feed pump fails. Whysoever your feed pump fails, you don't get any feeding to your cells. So quite not that good. Yeah? And what you will see now is how this model predictive controller automatically takes decision to recover the process. So to take action and to say, okay, if I now change that and that and that process parameter, I will still yield the same product amount at the end of the cultivation. This is what is the aim of this MPC approach here. So when looking at this, you see 5 different figures. On the left-hand side, the 3 figures are the input parameters, namely the feed volume, the temperature, and the inductor mass. So it's a microbial process. We are inducing this with IPTG. And what you now see is basically here, we have a pump failure. So we see no feeding anymore. And from the lines, I want to go through and help you. Basically, the blue line is the optimum offline condition. So the blue one is basically the process, how it should run. And then we see the green one is the no control group. So that means we have a pump failure, and afterwards we just continue as we did before. And the purple one is the MPC. So basically the tool that can automatically take actions. And you also see the control borders in which the MPC can take action. So you see that the MPC just doesn't do whatever he likes. We put some restriction of what the MPC is allowed to automatically do. And what you see basically that the MPC, until we are still on the ideal trajectory, it doesn't take any action. Okay, now here we introduce the pump failure at 7 hours, and then the MPC can take decisions to say, okay, there's something wrong. We have to do something about it. And then the MPC takes action, changes the temperature, and significantly changes the amount of inductive feed to the cell. So the MPC understood, okay, if we have less cells, we have to induce them more in order to get the same amount of product out. So this is a thing that the MPC learned from the data. We did not tell him or it per se. It learned this from the data, and it took the decision itself to control the process. And then once this pump failure was fixed, 3 hours later, then basically the MPC starts overfeeding. So significantly starts overfeeding, as you see from the top left image, that the trajectory of the feed is significantly increased. And by significantly increasing this trajectory of the feed, basically you can also see to the top right how the biomass catches up again. So while the biomass in the time where there was no feed really starts to decrease, not good, you can afterwards see basically how the biomass catches up again. And then at the end also overshoots the overall biomass concentration, because we reached the maximum feed at an earlier time. But what you can actually not see is the difference between the optimum offline control and the MPC, because the MPC takes action automatically, and you see that they are still overlapping. So what you can learn from this data is basically, or from this showcase, is that the MPC could maintain the same amount of product we were searching for at the end of the process, although we had a significant process interference, which comes from the pump failure. And that at the end is where we envision an MPC or a digital twin can unfold its full potential. So we use it for learning, we train faster, we find our optimum faster, and afterwards we take it for process control and monitoring. And this is basically the ultimate test for something like that, to say, okay, we have a change. What can we basically do? The limitation we have for sure is that only changes in the input parameter can be compensated. So if you have something completely different, which your model is not built on, modeling the unknown unknowns is difficult. Therefore, you will not be able to cope with that. And therefore, again, it's important to think what are likely events, which factors do I have to incorporate. And it's like always in life, the better you plan up front, the less issues you have afterwards. Last thing I want to empathize here is a project we're currently running. It's called eConti, where it's about to create a fully integrated, automated, autonomous biomanufacturing platform. And what we basically do is we want to build the first E. coli-based platform that should continuously produce protein A. In this case, it's just a showcase protein. We can replace this afterwards with other proteins. But the idea is here we have a cell line that due to the process, how we set it up with two chemostats connected to each other, we basically can overcome the problem of mutations over the time. And what you see is we have, after these two chemostats, we have a primary recovery, a filtration unit, and then a system from Tozo comes in, a multi-column chromatography system, and afterwards we basically get product out at the constant flow rate. And the way how we do this is basically we work together with Cubicon. They are collecting the data, and on Cubicon on top basically Novasign and Semvantage build on. So Semvantage is a company focusing on CFD modeling, while we as Novasign focusing on the whole end-to-end process. And we will basically build this MPC concept here in this whole unit operation. So we have only 4 unit operations, 2 upstream, 2 downstream. But at the end, the idea here and the vision is to fully control this process. And this is something we're currently working on. So what comes together is the physical world again with the digital world. So we want to digitalize this process, optimize it in silico, digital twin approach, and then go back and use it for control purposes. And this is exactly what is currently happening in this process. So now, in order to conclude my talk, whenever you talk about digital twins, it's important to consider first what a digital twin is and what it should deliver. Digital twins are anything and nothing. They for sure have a very high potential for the future. You just have to really consider what it should do and how you get there. In order to do that, consider the whole modeling workflow rather than just talking about a very fancy process modeler. So it's really about everything. The experiments are as important as the modeling approach. The hybrid models themselves, they're a very sound basis to build such a digital twin. So they can learn on only a little amount of data, so that's really cool. And the digital twin then can help you to optimize up and downstream. Today I only showed you upstream, but if you're interested, you can always reach out. We also have a lot of downstream applications. In order to save experiments, you should combine hybrid models with M and IDOE, depending on what you're aiming for. And at the end, to really make the most out of that, not just stop in the development department, take the digital twin along the whole process life cycle, and then use it as a model predictive controller, for example, at the end, by integrating it into a control system. With that, I'm at the end of my presentation. I want to thank you for your time. If you want to reach out, either reach me out on LinkedIn or send me an email. I'm happy to answer all of your questions. And I wish you a very productive conference and have a great day. Bye.